Swayam Prabha. Digital India. Educated India.
Welcome to my course on genome editing and engineering, and as part of module 5 on zinc finger nuclease technology, we are going to discuss today how we design zinc finger nucleases for genome editing. So, this lecture we will study about the design of the zinc finger motifs to bind to DNA targets of our interest. We already have studied about the zinc finger domains in the earlier lecture, where nature offers us many zinc finger motifs and which bind to diverse DNA sequences. And today we have the capability to modify those zinc finger domains and make them specific to our own needs. So, these we all started with basic research on zinc fingers, as well as the FOC1 endonuclease that we studied in the last lectures. And we know now that a ZFP or a zinc finger protein domain consists of 3 linked JF modules, and they bind to cognate DNA site and carries out activities like transcription. And FOC1 has a bipartite structure with a separable DNA binding domain and a non-specific cleavage domain. Now, put these ZFP and FOC1 knowledge together, a artificial enzyme can be constructed, which we call as zinc finger nucleases, and they are basically synthetic chimeric proteins which are composed of a specific zinc finger DNA binding domain linked to a non-specific FOC1 DNA endonuclease cleavage domain. However, overall these ZFNs bind to specific DNA sequences and cleave as per our requirements or desire. This is one of the glaring examples of the findings of basic research being taken to the translational stage, and this has revolutionized the genome engineering technology in a big way. So, you have come to know about Cinebas and Sakuran's work in the last class, and here he defined the functional domains of the FOC1 nuclease, and you can see this paper which was being published in PNAS, and this is the most important findings amongst others in this paper. The modular structure of the enzyme suggests that it may be feasible to construct chimeric endonucleases of different sequences by linking other DNA binding proteins. So, this is something very remarkable because you have a catalytic domain which is non-specific. So, if we attach that to other kind of DNA binding motifs, so we can engineer other nucleases with diverse specificities. Another work which is considered as a landmark work is the work by Pebble and Pebble in 1971. they found out from the crystal structure of the 3-finger ZIF268 bound to DNA that each finger used amino acid positions in -1, 3 and 6 of its alpha helix to contact a 3 base pair subsite of the DNA. So, this knowledge we are going to use extensively in the design of artificial zinc fingers. With all these findings, this seemingly simple mode of recognition of zinc fingers inspired many scientists to engineer zinc finger nucleases and create custom DNA binding proteins which can stimulate gene targeting at specific genomic loci in insect, plant and human cells. Using a linear array of several engineered zinc finger domains, many molecules were generated. Soon, this seemingly simple mode of recognition inspired scientists to engineer zinc finger nucleases to create custom DNA binding proteins which can stimulate gene targeting at specific genomic loci in insect, plant and human cells. Using linear arrays of several engineered zinc finger domains can be generated, and connecting the individual DNA binding modules with a highly conserved linker sequence also can be carried out, and it become possible to create polydactyl ZFPs that recognize long sequence of DNA with high specificity. For creating such polydactyl ZFPs, numerous platforms has been used like the modular assembly platforms. However, using these modular assembly platform turned out to be very tedious and complex in majority of cases due to the lack of reagents and protocols that permit rapid cross platform mixing and matching of the various zinc finger modules which were not easily available. Other problems included influence of adjacent modules in a polydactyl zinc finger which can have cooperative effects that affect activity of the entire DNA binding array. Wright and his associates have compiled a comprehensive publicly available archive of plasmids coding more than 140 well characterized zinc finger modules together with complementary web based software GFIT for identifying potential zinc finger target sites in a gene of interest. They also standardized the reagents on a single platform, enabling facile mixing and matching of modules and transfer of assembled arrays to express vectors without the need for specialized knowledge of zinc finger sequences or complicated oligonucleotide design. In addition, they have also described a bacterial cell-based reporter assay for rapidly screening the DNA binding activities of assembled multi-finger arrays. Screening with this protocol can be completed in approximately 24 to 26 days or less than a month. So, these are the various plasmids. You can see PC3XB ZF1 to 141. We have only listed here few for your understanding, and they have different IDs ZF1 to ZF141, and all these 141 molecules have identified different target DNA sequences ranging from GAA to RGA, and these are the ZF protein sequences. Most of these ZFs are 30 amino acid long. However, many shorter ones are also now reported, and you can visit this site by @genes for further information, and there is a spreadsheet containing information for all these 141 zinc finger plasmids as a CSV file which is downloadable. Now, let us move to the work of 3 scientists, namely Y.J. Kim, J. Shah and S. Chandrasekaran. In 1996, they reported the deliberate creation of novel site-specific endonuclease by linking 2 different zinc finger proteins to the cleavage domain of FOC1 endonuclease. So, they have not only found out earlier that the binding domain and the catalytic domain of FOC1 are in two separate domains, and if you divide them into two different domains, the catalytic activity is retained. So, to these catalytic domain, they joined zinc finger proteins or zinc fingers to create a new type of specificity. So, it became possible to create artificial nucleases that cuts DNA near a predetermined site. With all these sequence specificities at our disposal, today we can design so many different kind of ZFNs by using ZF motifs as per our DNA of interest and fusing those modules along with the nucleus domain of FOC1, and this was first pioneered by Sakuran and his group and published in PNS in 1996. So, what are the different zinc finger engineering platforms? So, briefly we have mentioned about the modular design of zinc finger proteins. So, zinc finger consortium established to ensure and to promote continued research and development of engineered zinc finger technology has enlisted 3 protocols which can be used for design of ZFNs. So, they are most basically the modular assembly method for engineering zinc finger arrays. The other is the CODA or the context dependent assembly method for engineering zinc finger arrays, and open or oligomerized pool engineering method for engineering zinc finger arrays. There are another method called the 2+2 strategy. This is however a proprietary platform and details are not available, and we will not discuss about this platform in this lecture. This is just to inform that apart from these 3, other technologies are also available. Let us first focus on the modular assembly. As the name suggests, we assemble the ZF modules one by one and then attach it to a nucleus enzyme. So, the first generation ZFP design entailed the use of a modular assembly in which individual zinc fingers were optimized against target triplet DNA sequences and linked together to form 3 or 4 ZFPs against 9 or 12 base pair sequences, because one module would bind to 3 bases. So, 3 will correspond to 9 and 4 will correspond to 12 base pair sequences. So, in such an approach, we join single zinc finger domains of known DNA binding specificities, and through these Wright and his associates contributed hugely for the development of this technology and the platform. While modular assembly provided examples of successfully applied ZFPs, Ramirez and his team raised concerns about unexpected failure rates for modular assembly of engineered zinc fingers in 2008. The high failure rate with this approach is likely due to the fact that they were ignoring the influence of neighboring zinc fingers on the sequence specificity of a given zinc finger. To overcome such deficiencies, selection guided assemblies were developed. So, Grisman et al. Described a method of gradually extending a new zinc finger protein across the desired 9 or 10 base pair target site, adding and optimizing one finger at a time. Briefly, the idea is to grow zinc finger modules using the first 2 zinc fingers of the GIF 268 trimer against a 6 base pair target to anchor a third zinc finger selected from a fuzz library using a new 3 base pair sequence as a bait. So, there are 2 rounds as you can see over here. So, this is the GIF 268 first and the second module, and the third module will be selected from this fuzz library. Okay. And after the first round, the second one is replaced, and after the second round the third one is also replaced, and the final construct will have a very high specificity. So, this process involve repetition of 2 additional times to replace each of the 2 remaining GIF 268 zinc fingers. The resulting synthetic zinc finger trimer can display high affinity and specificity towards the desired 9 base pair target sequence. In a similar approach, Jawang and his team used a Bacterial 2 hybrid system to optimize zinc finger binding to DNA sequences of interest. Next, let us move on to the little bit of discussion on the context dependent assembly CODA. Sander and his team described this CODA technique, a publicly available platform of reagents and software that is simple to practice and has a success rate for generating active zinc finger arrays comparable to that of selection based methods such as open. So, context sensitive selection strategies attempt to identify combinations of zinc fingers that work well together. Using CODA ZFNs, they rapidly altered 20 genes in various organisms like zebrafish, Arabidopsis and soybean. The simplicity and efficacy of CODA could enable broad adoption of ZFN technology and make possible large scale projects focused on multi-gene pathways or genome wide alterations. So, here in this cartoon depiction or figure, you can see the zinc fingers which are represented as colored spheres, finger 1, 2, 3 with 3 different colors binding to 3 different triplets, and then F4, finger F5 also binding to their respective sequences, and in both these you can see F2 is the common finger, and it is binding to his respective sequence TTG. So, here as already described, F2 is common to both the left 3 fingered array F1 to F2 F3 and the right 3 fingered array F4 F2 F5, and is positioned in the middle in both the triplets or trimers. The 3 base pair DNA subsites are represented as rectangles with corresponding colors. So, the 2 different 3 finger arrays which share a common middle finger F2 is used to create a 3 finger array with a new specificity by joining together the amino terminal finger F1 from the first array, the middle finger common to both arrays, and the carboxy terminal finger F5 from the second array. So, due to this fusion, this F3 and F4 are removed, and these combination of F1, F2, F5 now gives a new kind of a specificity. This modular assembly strategy treats ZFs as completely independent units. Cornu and associates have found that CODA, in contrast to the modular assembly strategy, yield multi-finger domains that show high activity and low toxicity as zinc finger nucleases in the human cells. These context sensitive selection strategies account for potential context dependent effects, including cooperativity of zinc finger binding and occasional recognition of a 4th base in the target sequence. Let us now move to the next platform, which is the oligomerized pool engineering or OPEN. Modular and his team described OPEN, oligomerized pool engineering, which is an open source combinatorial selection based method for engineering zinc finger arrays for developing ZFNs. OPEN method has been found to have a higher success rate than previously described modular assembly methods for engineering ZFNs. Open selections are carried out in E. coli using a bacterial to hybrid system, and they do not require specialized equipment. So, open relies on an archive of pre-characterized zinc finger pools that are organized based on their binding specificity to a given 3 base pair sequence. After identifying the desired genomic target, the appropriate mixtures of zinc finger pools are then randomly assembled using overlapping PCR and screened for the zinc finger assembly displaying the most potent binding activity to the desired 9 base pair target. So, here we have 3 archives 1, 2 and 3. So, we carry out PCR and assembly of the 3 modules, and we go for selection by 2 hybrid methods and obtain the desired 9 base pairs at the end of this process. With open resources can generate multiple customized zinc finger nucleases in about 8 weeks or 2 months, but the labor and expertise required to screen community libraries have limited its broad adoption. So, this figure, if you observe, we have a histec in one end, and then we have the zinc finger triplets over here, as you can see, and then this is the linker which join the ZF domains to the functional domain, and then these are the specific restriction sites in this construct. So, basically this is used for the production of hybrid enzyme zinc finger nucleases. This is a map of the zinc finger FN fusion gene construct which produced hybrid enzyme ZFN when placed downstream of T7 promoter in an expression vector. PCR was used to construct expression vectors of the zinc finger fusions. The programmable nucleases take advantage of natural cellular pathways of DNA repair for the introduction of targeted sequence changes. And we have discussed in the earlier lecture that the dimerization of the DNA cleavage domain is very important, and we have also discussed the involvement of the various residues or structural components in this dimerization. And once that dimerization is successful, the ZFNs will create double strand breaks. Okay. So, now it may follow 2 pathways as we have discussed in the beginning of our course. It can lead to non-homologous enjoining, and there can be some targeted mutagenesis over here, or it can follow the homologous recombination pathway, and we may add a donor DNA over here which will lead to the targeted genetic replacement. So, we are going to use the knowledge of zinc fingers and Foquan nucleases to create fusion proteins having domains of zinc fingers and the catalytic DNA cleavage activity of the Foquan enzyme, and then we are going to deploy them inside a cell where they will carry out double stranded DNA breakage, and then we may direct the repair either in the non-homologous enjoining pathway or in the homologous recombination pathway by other interventional steps, and thereby carry out gene editing which may be simple point mutations or targeted gene replacement. The current methods to manipulate plant and mammalian genomes have two limitations: the very low rate of homologous recombination at the targeted site and the relatively high rate of random non-targeted integration elsewhere in the genome by non-homologous adjoining. In these cells, homologous recombination occurs at a very low rate compared to the NACJ. For most mammalian cells, targeted recombinations via homologous recombination are overshadowed by nearly 1000-fold higher random non-targeted integrants. Cells use the universal process of homologous recombination to maintain their genomic integrity, particularly in the repair of double strand breaks, which otherwise would be lethal, and these are important facts we have emphasized in the last lectures. The repair of these DSB in a damaged chromosome by HR is a highly accurate form of repair, which uses the homologous DNA from the undamaged chromosomal partner as a template. Gene targeting is the process of modifying a gene by homologous recombination, which uses an extra chromosomal fragment of donor template DNA and invokes the cell's homologous recombination machinery for sequence exchange. Gene targeting is not a very efficient process in plant and mammalian cells. Only 1 in 106 cells provided with excess template sequences undergo the desired gene modification. However, when a targeted genomic DSB is introduced in cells, it induces homologous recombination at that local site to repair the DSB in a larger fraction of the cells compared with spontaneous homologous recombination. So, for generation of specific desired genomic DSB, which is the rate limiting step in homology directed repair technology for gene modification, we will use zinc finger nucleases, other similar nucleases. So, they are also called as the molecular scissors for genome surgery. A schematic representation of a pair of 4-finger gelaphens bound to recognate sites can be seen in this figure. There are 4 members over here, fingers, and these are the nuclease domains. A pair of gelaphens require 2 copies of the 2 base pair sequence recognition sites in a tail to tail orientation. They effectively have a 24 base pair recognition site for these catalysis to happen, which is long enough to specify a unique genomic address in human cells. So, the off target cleavage is highly reduced due to this high or long recognition site involvement. So, if they are triplets, the requirement will be actually 9 plus 9 or 18 base pair recognition sites. Since the gelaphen binding sites in the genes are not identical, both gelaphens that bind these sites need to be introduced into the cell to induce a targeted genomic DSBs. So, that is another advantage of this technology. So, we need to design for both the forward and the backward, or sense and the antisense strands, and because these enzyme can act only if it is dimerized. So, if the binding on any of the strands, either the sense or the antisense fails, the cleavage will not happen. So, this gives additional specificity to such a constructs. The gelaphen sites in the genes are separated here by around 5 base pair as shown in the schematics. So, single gelaphen array versus dimeric gelaph nucleus sites. So, here you see a single gelaph array comprising of 3 fingers binding to one of the strands. So, gelaph finger protein bound to a single gelaph array target site as shown in this picture. A single gelaph array target site consisting of 3 to 8 adjacent DNA triplets or 9 to 24 nucleotides on the same strand of the DNA. Each triplet of DNA is recognized by one finger or ZF module. The cartoon illustrates an array with 3 fingers bound to a single ZF array target site, in this case a 9 nucleotide DNA. So, here is the single ZF array versus dimeric ZF nucleosides, and here you have 2 partners. The single the zinc finger nuclease bound to a dimeric ZF nucleus target site is being depicted in this figure. A ZF nucleus target sites consisting of 2 ZF array sites on a complementary DNA strands separated by a spacer of around 5 or 6 nucleotides, and in this configuration fork1 endonuclease monomers in blue covalently linked to the C terminal end of each ZF array domain forms an active dimeric nuclease and induce a double stranded break in the spacer DNA between 2 ZF array binding sites. So, these active dimeric nuclease is very important for the cleavage of DNA. Here, due to absence of its partner on the other strands, the enzyme is not active and it is not able to cleave. So, this concept is very important to understand the mechanism by which zinc finger nucleases operate. So, let us now study about ZFN mediated gene targeting. Let us now study about ZFN mediated gene targeting. In the earlier slide, we discussed about the requirement of dimerization of the nucleus domains to induce a double strand break. So, we are going to use that double strand break generation by ZFNs for ZFN mediated gene targeting. So, there is a gene X as you can see in this picture, and then we expose it to zinc finger nucleases, which will generate a double strand break in this gene because the zinc fingers that we have designed bind to the sense and the antisense strand, and the ZFNs dimerize to generate this double strand break. Now, due to some kind of deletion events, there may be some kind of mutation occurring here, but this pathway comes into action over here, and then there is a mutagenic repair due to NSEJ, non-homologous end joining, and we get a pool of mutants with insertion or deletion in this case. Now, similarly, if we add engineered zinc finger nucleases, and we also add some mutant donor templates, so there will be template dependent repair in this case, which is due to the homology directed repair, and then we get here a specific mutant which is directed by the donor template. So, this is the strategy by which we use the ZFN mediated gene targeting. This ZFN mediated gene targeting involves various steps as follows. Number 1, you need to deliver ZFNs alone to induce a targeted DSB in gene X of normal cells and stimulate NSEJ to generate a pool of mutants, some of which will be frame shift mutations resulting in functional deletion of gene X, that is knockouts of gene X. Alternatively, we deliver ZFNs and the mutant gene X donor fragment into normal cells to induce a targeted DSB and stimulate homologous recombination to generate a specific gene X mutant, that is knockouts or knock-ins of gene X as the case may be, to achieve gene editing. Deliver ZFNs and the correcting donor gene X fragment into the mutant cell. The second step is to monitor for gene correction or mutagenesis at the targeted gene loci as per the investigator provided donor gene X template, and it is also critical to show that the donor DNA fragment has not integrated elsewhere within the genome of the cell by using Sakuran blot. So, this has to happen exactly here and not somewhere else, and that has to be confirmed. So, it is an important step in the ZFN mediated gene targeting. ZFN mediated gene targeting in human cells, let us study a bit about it. Targeted ZFN mediated disruption of the gene X by NSEJ occurs when cells are transfected with designer ZFNs alone. The ZFNs induce a targeted genomic DSB in cells which stimulates non-homologous enjoining, that is mutagenic by nature. It gives rise to a pool of different gene X mutants. Let us now discuss about ZFN mediated gene targeting, which involves various steps as followed. And the first step is to deliver ZFNs alone to induce a targeted DSB in gene X of normal cells and stimulate the non-homologous enjoining to generate a pool of mutants, some of which will be frame shift mutations resulting in functional deletion of gene X, that is gene knockouts of gene X. Alternatively, deliver ZFNs and the mutant gene X donor fragment into normal cells to induce a targeted DSB and stimulate homologous recombination to generate a specific gene X mutant, that is knockout or knock-ins of gene X as the case may be. To achieve gene editing, we need to deliver ZFNs and the correcting donor gene X fragment into the mutant cells. Once this is done, we need to monitor for the gene correction or mutagenesis at the targeted gene loci as per the investigator provided donor X template. And it is also very important to determine that the donor DNA fragment has not integrated elsewhere within the genome of the cell by using Sakuran blot. So, identifying ZFN target sites near the targeted locus within the gene X of interest is the first most important step in this procedure. Next, in this procedure we go for designing and or selecting zinc finger proteins that recognize the Susan ZFN target sites, and then we convert the designed and or selected ZFPs into ZFNs by attaching it or covalently binding it to the nucleus domain. So, this is a resource site from where you can get many of the reagents required, and you have these various platforms as well here, the open and the modular assembly engineering reagents, and we have discussed about these various platforms earlier. Numerous research groups have experimentally determined zinc finger modules that bind many of the 64 possible DNA triplets. A couple of web based tools that facilitate the design of zinc finger proteins that can bind to specific DNA sequences are also available. These tools are most often based on a widely employed method of ZFP design, the modular assembly approach, in which pre-existing individual zinc fingers are linked together to recognize desired target DNA sequences. GFIT leverages the combined capabilities of 3 of the largest and best characterized module archives by enabling users to select fingers from any of these sets. GFIT searches a query DNA sequence for target sites for which a ZFP can be designed using modules available in one or more of the 3 archives. In addition, GFIT output facilitates identification of specific zinc finger modules that are publicly available from the zinc finger consortium. This is GFIT is freely available from this site, and you can download and you can use it or experiment with it and try to generate some zinc finger nucleases, maybe just for fun. So, this is how this website resource looks. This is a software package and which is designed to aid research and application of gene editing and expression technologies. So, these identifies potential target sites in DNA sequences for several DNA binding platforms using publicly available reagents and protocols, including for CRISPR, Cas and tails, and of course, GLFPs. We are going to discuss about these other two technologies soon, one by one. Now, how do we deliver the zinc finger nucleases inside the cell? We now have the capability to design GLF modules by various platforms, and you also know about software platforms available which can be used to design effective zinc finger modules, and then we can attach those to nucleases and generate ZFNs of our interest and specificity to bind to DNA targets of our choice. Now, once we do that, we need to deliver it inside the cell, so that it goes inside the cell, bind to the specific DNA sequences, and then generate double stranded breaks and allow the cells DNA repair mechanisms to take over and give the desired results, whether it is a point mutation or gene knockout or gene knock-in. So, there are very different kind of delivery methods used for gene delivery. We have viral vectors like AAV, ADVs and LVs, or lentiviral vectors, adenovirus vectors or adeno associated vectors. Then we have, so you can see here the different viral vectors being used in this case. Then we have non-viral vectors which may be polymers, liposomes, etc., or cell penetrating peptides, CPPs. Then we have other methods, physical methods, which may be microinjection, electroporation, etc. So, these in brief constitute the three major type of gene delivery platforms or methods. So, in vivo delivery systems for zinc finger nucleases and their expression cassette. So, we use virus, as we already told in the earlier slide. The virus life cycle involves infection and replication. In infection step, the virus enters into target cells after recognition and release the viral genome for replication. In replication step, the progeny are released outside cells after synthesizing the viral genome copies into cells. Following these, fresh infection steps in nearby cells happen or circulation begins. This is useful in using virus vectors to encode and deliver genome editing programmable nucleases to target tissues or cells and achieve genome editing therapy. So, one of the earliest viral vectors used was the adenoviral vectors, or the first generation adenoviral vectors, which are obtained by substituting the gene E1 or E1 and E3 followed by the second generation ADVs, which lack more than 2 early genes including E2 and E4. Compared to the first generation, the second generation ADVs provide an extended genome packaging capacity. However, it is limited by the inflammatory response of the host and the inability of replicating in vivo, including its cytotoxic T cell response. A systemic delivery of ZFNs against HIV infections has currently entered clinical trials. The expression unit coding the right and left ZFNs is inserted into a serotype 5 ADV pseudotyped with serotype 35 fiber. The ADV ZFN system is designed to repair the autologous CD4 plus and helper T cells from HIV infected patients. Another virus that is used are the lentiviral vectors. The retrovirus can spontaneously penetrate the intact nuclear membrane. For this, lentiviral vectors are now commonly used for in vivo delivery in genome editing therapy. As the delivery vector of ZFN's technology, lentiviral vectors which can accommodate sequences up to around 10 kilobases or theoretically allow for site specific genome modification or addition in predefined genomic sites. However, they have been avoided because of multiple tragedies involving patient death in earlier clinical trials. Therefore, advanced types such as integrase defective lentiviral vectors have been heavily explored in the recent years. Another viral vector that is used in delivering ZFNs for genome editing is the adeno-associated virus vector. The adeno-associated virus vectors are the commonly used delivery vector in genome editing technologies for its potential site-specific integration ability and low immunogenic characteristics, unlike the others discussed earlier. Some examples of these are isocohedral non-enveloped viruses in the Dependovirus genus of Parvoviridae family. However, it also has certain limitations. The major limitation of AAV vector system is their relatively small genome size, around 4.7 kb, which restricts the genome engineering nuclease complex that they can carry. However, with careful design, sequences for expressing ZFNs, each sequence is around 1 kb, and an optional donor DNA template can be encapsulated well by the AAV vectors. We can also deliver ZFNs directly. So, for a protein to be delivered directly, overcoming the barriers in the cell membrane, penetration by proteins due to their low lipophilicity, pH sensitivity, and degradation by endogenous proteases, we have to devise certain strategies. Fortunately, ZFN proteins can penetrate cell membranes due to the positive charge of cis2 his2 zinc finger domains, and it has been found that the direct delivery of ZFN proteins can disrupt the CCR5 gene in both HEC 293 HDF cells and human CD4 plus T cells. Increasing the stability of ZFN proteins is vital or critical to achieve the direct delivery of ZFN proteins, because ZFN proteins will be completely degraded in within 4 hours. One of the possible strategies to keep ZFN stable is to modify the lysine residue needed in the degradation of the protein. So, these are in brief the typical in vivo delivery systems and candidates for genome editing nucleases and their expression cassettes. You can see here the typical delivery systems, the viral system AAV, ADV, HV ADVs. Then you have here others not important for ZFN. Mostly the AAVs are used for ZFNs as well as CRISPR-Cas9, and ADV is also being used over here. And this slide is important from the point of view to understand the status of the clinical trials of these particular delivery platforms. Here information is not available, or they are not being put forward for clinical trials. But here you can see that the ADVs, the clinical trials, one has been completed, as well as clinical trial phase 2 has also been completed. There are certain advantages of this. They are low, they show low off target mutagenesis, but the problem is the immunoreactivity and also the cost currently. Let us go to the next section of this lecture where we will discuss about the design strategy of gel defense. So, first let us have a small discussion on fusion proteins, which is maybe very familiar to you. Particularly we use many fusion proteins by fusing the gene green fluorescent protein to it, so that we can trace the location of the targeted protein by observing the fluorescence of the green fluorescent protein. So, a fusion protein is an artificial protein which is constructed by joining at least two domains of individual proteins that are encoded by separate genes. And for that matter, ZFN is a fusion protein in a way. It is joining the zinc finger domains with a nucleus or catalytic domain of FOC1. Such a fusion gene is transcribed and translated as a single unit producing a single polypeptide. Fusion proteins can also occur in vivo due to chromosomal rearrangements. They are not just engineered all the time, they may happen due to natural chromosomal rearrangement due to any of the mechanisms, cellular mechanisms, inherent cellular mechanisms. So, okay, this is the mRNA of a fusion gene. We have gene 1 here which is contiguous, continuous to gene 2, and then as it reaches through the ribosome for the translation process, we get a product of two proteins which are covalently bound to one another. The fusion protein product, you can see this is 1 plus 2. The two domains may carry out two different continue to carry out the independent functions which was there in the native protein. So, there is another important concept that we need to remember or learn at this moment. Those are the 2A peptides. So, what are these 2A peptides? So, these 2A peptides are 18 to 22 amino acid long self cleaving peptides. They induce ribosomal skipping during translation of a protein. We need to remember the term ribosomal skipping. Okay. So, let us go back to the earlier slide. You can see mRNA becomes a protein only after translation that happens in the ribosome, and we have a term here ribosomal skipping, which is related to the word ribosome. 2A peptides share a core sequence motif of DX, EX, NP, GP and are named after the virus in which they have been first described. F2A is the first described 2A peptide derived from foot and mouth disease virus, which occurs basically in bovidae or for example in cattle. The name 2A comes from the gene numbering scheme of this particular virus. So, F2A basically derived from foot and mouth disease virus. Others like P2A is derived from porcine tascovirus, and T2A is derived from Thossia asigna virus. So, we have at least say 3 different peptides which can induce ribosomal skipping, namely the 2A peptide known as the F2A peptide or P2A peptide or T2A peptide. All these assist directly in synthesizing polyproteins by causing the ribosome to fail at making a peptide bond. What we mean by this we will learn very soon. So, we have seen this construct of two genes, gene 1 and gene 2 to create a fusion protein. So, they are continuous one after the other, but in our current scheme of things we are going to introduce these F2A peptide particularly in between these two genes. And by doing this, we are going to have a very different kind of an outcome. The introduction of these 2A or F2A sequence will lead to ribosomal skipping. What do you mean by ribosomal skipping? This is a mechanism of translation in which a specific viral peptide prevents the ribosome from covalently linking a new inserted amino acid and let it continue translation, resulting in co-translational cleavage of the polyprotein. However, the molecular mechanism of this 2A peptide mediated cleavage is still not very clear. There is a probable explanation given forward by some of the scientists. The cleavage is triggered by skipping the formation of peptide bond between the proline and glycine in C terminal of 2A peptide. The peptide located upstream of the 2A sequence will have extra amino acid on its C terminal, and the peptide located downstream the 2A sequence will have an extra proline on its N terminal end. So, this is the fusion protein of gene 1 and gene 2, which has a 2A intervening, separated by a 2A intervening sequence. So, when translation takes place of these mRNA, the first protein is produced, okay, and moment the ribosome come in interaction with the 2A sequence, there is a skipping or ribosomal skipping happening, as a result of which the protein 1 will be released. But the translation continues beyond 2A sequence, and it goes on to produce the second protein. So, by this method of inserting a 2A sequence in between the sequence of 2 genes, mRNA sequence of 2 genes, we can produce 2 separate proteins in a single read. And if we do not have that sequence, we will have a fusion protein, because there is no any 2A here. But in the case where 2A is introduced, we get 2 different proteins. So, this strategy, design strategy is very, very important for ZFN design. We go back to the fact that the endonuclease domains of Focon must form dimers to cleave the double stranded DNA sequences. So, ZFNs are required in pairs to bind to the target sequence, and we have also discussed a single ZFN molecule is not going to cleave the DNA. We need a partner which bind to the complementary strand. So, these binding of 2 ZFN molecules in the opposite orientation with the correct spacer is very, very important for double strand breaks. So, to provide 2 ZFN molecules inside the cell, we may clone them into separate vectors, but the success rate would be low, because if one of the vectors do not enter, or efficiency is low, or for any other reason the vectors are delivered, but one of the constructs ZFNs are not expressed properly, the dimerization is not going to happen satisfactorily, and the double strand break generated will be low. To avoid that, we will put both the ZFNs into one single construct and separated by a F2A sequence. We cannot generate a fusion protein, because that is not going to be helpful to us. So, this will ensure that both the ZFNs are delivered inside the single cell, and they produce both the nucleases which will bind to the specific targets and after dimerization they will form the double strand breaks and allow the desired mutagenesis to occur. So, the introduction of the two-way sequence helps us in cloning them together and ensure the co-expression as well as the double strand breaks. So, here is a schematic representation of different constructs of baculovirus vectors. So, number 1 you have carrying either of the ZFNs. So, you have the ZFN-L and ZFN-R, and you can now understand what is this F2A sequence is all about. So, ZFN-L may be binding on one of the strands and the ZFN-R will bind on the other strand. So, this CMB is the human cytomegalovirus intermediate early gene promoter. ZFN-L is the zinc finger nuclease targeting the left flank of the CCR5 gene. This is important gene in the pathogenesis of HIV, which we are going to discuss in the applications part. The zinc finger nuclease ZFN-R is the ZFN targeting the right flank of the CCR5 gene, and this F2A is a two-way sequence derived from the foot and mouth disease virus, and these are the homologous regions of the DNA sequences, and this is a polyadenylation site, and in certain other constructs we use the GFP, and you have the LTR, which are the long terminal repeats. So, to sum up briefly, we can have various constructs of baculovirus where we carry the ZFN right and ZFN left sequences, intervening sequences with intervening F2A foot and mouth disease 2A sequences, and we know that this is being done to ensure that both the ZFN molecules are delivered together and expressed together to carry out efficient double strand breaks and the targeted mutagenesis. Thank you for your patient hearing.